Between chemistry and life lies a shadowy frontier, the world of viruses. They have no metabolism, no heartbeat, and no independent will. Yet, in the presence of a host, they awaken, multiply, and evolve. Viruses exist at the edge of definition, blurring what it means to be alive. The story began in 1892, when Dmitry Ivanovsky studied diseased tobacco leaves. He filtered the sap through fine porcelain designed to trap bacteria, but the clear liquid still caused infection. Soon after, Martinus Beijerinck described it as a contagious living fluid, a new form of life, too small to be seen, yet capable of reproduction. The invisible parasite had entered science. For decades, viruses were known only by their effects: disease, deformation, decay. It wasn't until the 1930s that the electron microscope unveiled their form. Under its beam appeared crystalline structures of perfect symmetry, icosahedral shells, helical coils, geometric simplicity wrapped around genetic code. Each virus was little more than a message in molecular form, a script awaiting a reader. That reader was the host cell. Outside the cell, a virus is inert. Inside, it becomes alive by proxy. It docks, injects, and rewires. The host's ribosomes, enzymes, and membranes turn into viral machinery. In hours, hundreds of new particles emerge, ready to begin again. Where then does life truly reside? In the virus, the cell, or their temporary union? Bacteriophages, viruses that infect bacteria, helped answer that question. In the 1950s, Esther Liederberg discovered phage lambda, a virus that could either destroy its host or hide within its DNA. That duality, lysis or latency, became a model for genetic control. Phages taught biology how genes switch on and off, how memory can be molecular, and how evolution balances conflict with cooperation. As research expanded, viruses proved astonishingly diverse. Their genomes may be RNA or DNA, single or double-stranded, linear or circular. Some are smaller than the simplest genes. Others, like mimivirus, are larger than bacteria, carrying hundreds of genes once thought unique to cells. Each new discovery blurred the boundary between the living and the non-living. And viruses are not merely agents of destruction. They regulate ecosystems by culling microbial populations and recycling nutrients. In oceans, they shape plankton communities that drive global carbon cycles. Even within us, ancient viral genes have become part of our own genome, relics of infection that now guide embryo development and immune defense. We are, in part, the descendants of viruses that learned to stay. Today, these same entities offer tools for medicine. Engineered viral vectors deliver healthy genes to damaged cells. Oncolytic viruses are being designed to target cancer. Humanity has begun to harness its oldest enemies as allies. To study viruses is to confront life's threshold. They are neither alive nor dead, yet they define the boundaries of both. They rewrite genomes, reshape ecosystems, and remind us that life is not a fixed state, but a continuum. A conversation between chemistry and replication. At that frontier, one truth persists. Existence begins not with consciousness or complexity, but with the ability to leave a trace, to copy, to persist, to whisper through molecules, "I was here."